Hi everyone. Today you are going to perform Activity 4, which is entitled Cell Counting and Cell Viability. Cell counting is defined as any of various methods for counting or similar quantification of cells, including medical diagnosis and treatment. This process is actually used in both medical and research laboratory in subculturing subsequent cell-based assays, especially if you are going to perform serial dilution, say for example, and at the same time assessing physiological response of cell to external factors. For the cells that are usually counted using haemocytometer, we have of course the blood cells, which includes the red blood cells or the erythrocytes, the white blood cells, which includes the monocyte, the lymphocyte, and of course the neutrophils, and your platelets, which are the thrombocytes. Aside from that, we also have bacteria in the laboratory. You usually count the number of E. coli. Also, of course, the number of Bacillus species, which includes Bacillus thuringiensis or Bacillus subtilis in the process. We also have yeast, which you will be counting today, which is of course again the Saccharomyces cerevisiae, and the number of spores which may be associated with that of the bacteria or that of a fungi. So if we're going to perform the counting of your organisms, let us first talk about these organisms, especially of course the culturing of your yeast. Yeast are also known as your Saccharomyces cerevisiae. Yeast are of course eukaryotic single-celled organisms, and you know for a fact that the size of these organisms are around 3 to 4 micrometer. In the lecture, we usually say that less than 10 micrometer are usually associated, of course, with that of bacteria, correct? That's 1 to 10 micrometer. And 10 micrometer to 100 micrometer is associated with that of eukaryotic organism. But be reminded that your Saccharomyces cerevisiae are, of course, again classified as your eukaryote. For reproductive structure, the reproduction of your yeast are usually associated with budding, although again, of course, others through binary fission. It can ferment sugars for ethanol production, which we usually do because we use them for fermenting different fruits to make, of course, wine, and then later on maybe vinegar after some time or by the use of another organism. And the optimum temperature for yeast would be at around 25 to 30 degrees Celsius. The optimum pH is at 4.5 to 6 and commonly cultured in YPD. YPD media is known to be yeast extract, peptone, dextrose. So it's a combination of the yeast extract, the peptone, and of course the dextrose in a media. So you can actually have this in the solid or the liquid media. Aside from that, for the microbial growth curve, you can see that there should be, of course, different parts, 4 parts specifically for the microbial growth curve, which includes the lag phase, the log phase, stationary phase, and death or decline phase. What is the difference between the log, the stationary phase, and the decline or the death phase? Well, in the lag phase, there is no increase in number of living bacterial cell. Why is there no increase in this phase? Because this is actually the time where you introduce the organism to that of the media. And because the organism are starting to assimilate the nutrients coming from that of the media, in this time specifically, there is no growth associated with that of the organism. However, as you proceed, and while they assimilate, they get into the nutrients and other substances found in that of the media, your cells will be able to undergo cell division now. And when they undergo cell division continuously, there will be an exponential increase in number of living bacterial cells. However, sometime in the process, you will be encountering the stationary phase where there is plateau in number of living bacterial cells and the rate of the division, and that is roughly equal, which means that because you have been undergoing cell division for quite some time, there will be an increase now of toxic substances and decrease in the nutrients associated in the cell. And because of that, there will be an stationary phase. My professor usually says that during the stationary phase, hindi naman talaga siya dire-diretso lang. This parang zigzag siya. Why? Because there will be an increase and then there will be decrease, increase, decrease, and so on and so forth. But if you're going to look at that, really, there is actually the formation of a plateau in the growth of your organism, and that is why we call it stationary phase. For the death phase or the decline phase, there is an exponential decrease now in the number of living organism, which means that the organism, because of the amount of the toxic substances and the decrease in the availability of nutrients in the media, the organism will of course die, and in that case, they experience death or decline phase. To determine, therefore, the number of organism in your culture, may that be, of course, the whole number of organism or the living organism alone, you will be using the haemocytometer. Haemocytometer came from the word hemo, which means blood, cyto, which means cell, and meter, which is of course measurement or counter. That is why haemocytometer is usually, before, haemocytometer is defined as an instrument to count blood cells specifically. But now, can we use haemocytometer in counting other organisms such as, again, bacteria or yeast or spores and others? Yes, we are using haemocytometer. And specifically, when we talk about haemocytometer, we will be using Neubauer's slide. Neubauer's slide is a thick glass slide with an H-shaped groove in the center, as you can see in the figure. Scales are found on the side of the central, horizontal to that of the center, and of course the depth of the scales is 1/10 or 0.1 millimeter. So you can see here the presence of the haemocytometer, specifically the Neubauer's slide. For your haemocytometer or the Neubauer's slide, if there is a cover slip added to that of the Neubauer's slide, that specific part or chamber is actually what we call now the Neubauer's chamber. So if you're going to look at the figure, you have of course the presence of a gap which is equal to 0.1 millimeter high between the cover slip and at the same time that of the Neubauer's slide. Aside from that, you have of course the haemocytometer and the finely ruled grids. Let's focus our study with the finely ruled grids. This is the grid of your haemocytometer, and if you're going to look at it, this is subdivided into 9 quadrants. So we have 1 on top, 2, 3, and then in the middle 4, 5, 6, and then at the bottom we have 7, 8, and 9. This is actually all in all 3 by 3 millimeter, because each of the quadrant is equal to the big quadrant, or the big squares is equal to 1 millimeter by 1 millimeter. So all in all, that's 3 by 3 millimeter, or 9 big squares or quadrant. You can see here, however, that each quadrant is subdivided into, of course, smaller squares. As you can see, you have there 1, 2, 3, 4 by 4 squares. In the center, you have there 5 by 4, but in the center, so in quadrant 1, if we're going to look at it, that's 4 by 4. In quadrant 3, we have 4 by 5. In quadrant 2, rather, 4 by 5. But in quadrant 5, specifically in the center, we have a 5 by 5 millimeter quadrant. Sorry, not 5 by 5 millimeter, but 5 by 5 squares. So we have 5 squares on top, and at the same time 5 squares at the bottom. So for haemocytometer, if you're going to count that, you are going to count that per quadrant. So one will be, you might want to count the number of cells in quadrant 1, or quadrant 3, or quadrant 5, or quadrant 7, or quadrant 9, and so on and so forth. But we have, of course, a rule for counting the number of cells in your haemocytometer. And what is the rule? The rule is that you are not going to count cells touching the bottom line and the right line. So, bottom line and the right line. Why are you not going to do that? Because your movement is from left to right, correct? Left to right, then going to bottom, and then right to left, go down, and then left to right, and so on and so forth, until you finish counting the whole quadrant. Now, if you're going to that of the right, you should not again count the number of cells to that of the bottom line. Why again? Because if you're going to move, you might forget that you were able to count the cells already. And just to be sure that you have not counted the cells yet, you don't count the cells touching the bottom line and the right line. And then again, so this is to avoid double counting of cells in your haemocytometer. So say for example here, if you're going to count the number of cells specifically, we said that do not count the number of cells on the right and on the left, correct? On the right and at the same time the bottom line. So in that case, how many cells do we have here? Do you count in this haemocytometer specifically? So that's 1, 2, 3, 4, 5, 6, 7 all in all. Because that one cell that is on the left should be already counted, and in that case again, this is composed of all in all 7 cells. Let's talk about your activity this time. At the end of the activity, the students should be able to, first, you should be able to measure the optical density of cells in liquid culture using spectrophotometry. We already made use of the spectrophotometer in our previous activity, and in that case, I believe that you will be able to perform this part of the activity. Next, perform serial dilutions and yeast cell counting using the haemocytometer, and determine the cell viability by trypan blue exclusion technique. In the growth curve of your yeast cells, we will be, of course, providing you a culture of Saccharomyces cerevisiae. Sample 1 ml and place in the microcentrifuge tube. Determine cell density by reading 900 microlit of sample in a spectrophotometer at optical density of 600 and return the culture to the incubator. So, what does this mean? You will be, of course, getting 1 ml of the culture. However, you are going to put the 900 microlit of that sample to that of the cuvette, and you're going to check that in the spectrophotometer at optical density of 600. And you are going to do it every after 40 minutes until the end of the laboratory period, which means that do the spectrophotometry and then put it in the incubator, get it, do the spectrophotometry, put it back in the incubator, and do again the activity until the end of the laboratory period. For cell viability assay, you are going to clean the haemocytometer and cover slip first with 70% ethanol, and then add 100 microlit of 0.4 trypan blue to microcentrifuge tube containing cell suspensions taken at different time points. Mix well by pipetting. This means that this is actually connected to that of your activity in your cell counting associated with spectrophotometer. So take 20 microlit, only 20 microlit of the mixed sample and introduce to the grid in the haemocytometer. Place cover slip over the raised glass rails and observe under low power objective. How are you going to do that? Member 1 of the group will count the number of cells in quadrant 1. Member 2 will count the number of cells in quadrant 3. Member 3 in quadrant 5. Member 4 in quadrant 7. And member 5 in quadrant 9. Once you are done counting the number of cells in each of the different quadrants, you are going to perform, of course, this part of the activity, which is to determine the concentration of viable cells per ml from the formula where viable cell count is equal to the number of viable cells over the number of large squares counted times dilution factor of 10 raised to 4. Dilution factor is usually 2. That means 1:1 dilution with trypan blue. So 1 portion of your sample and 1 portion of your trypan blue. And of course, that's 10 raised to 4 is the conversion factor. And when you determine the percentage viability by the formula, viability percent, total counted cells minus the total counted dead cells over the total counted cells, which means that you are going to count separately the number of the cells that are not stained and the number of cells that are stained, because you will be able to identify specifically which is actually alive or living cells and which are considered the dead cells. And in that case, again, you will be able to get the percent viability. But of course, all in all, you will be able to identify, correct, the number of cells specifically in each dilution in each of the different time intervals. So if you have questions with regards to the use of haemocytometer, you can just approach me or Dr. Chavez or any of your course facilitator in this matter. So if you have questions, again, send to us your questions. and we will be glad to answer them. Thank you very much, everyone, and enjoy your activity.